Since the first successful bone marrow transplant in 1956, there has been more than 1.5 million transplants globally. These transplants are performed with curative intent. However, these patients do have a high risk of premature morbidity, and often that can have a long latency period. A recent study showed that approximately 40% of patients that have a bone marrow transplant, if they survive the first 2 years, have a chance of premature morbidity in the following 15 years, meaning that these patients need to remain heavily engaged with the healthcare system for lifelong monitoring. One method of monitoring, looking at engraftment of donor cells, confirming that via chimerism, is what we're going to talk about today. This testing is done generally on a quarterly basis. It can be done on blood, bone marrow, or sorted cells, or a combination of these different cell types. Chimerism monitoring enables early detection of disease relapse, the level of engraftment, graft versus host disease, tumour versus host interaction, facilitating therapeutic interventions. The current method for chimerism monitoring is an analysis of short tandem repeats. This is a quite labour-intensive methodology, particularly in the analysis stage, making it very difficult to increase throughput to match the growing demand for this type of testing. My name is Darryl Irwin, and I am the Senior Director of Scientific Affairs at Agena Bioscience. And today I'm going to introduce to you Agena's new Chimeric ID panel, a panel that has completely automated data analysis, meaning that it's scalable with your increased demand, but also is reduced cost because of that reduction in labour. And of course, it has the accuracy and sensitivity that you've grown to expect with STR for this type of analysis. Some recent studies have shown that the number of hematopoietic stem cell transplants in the United States is approximately 20,000 transplants per annum. And in Europe, it's approximately 40,000 transplants. Of these, 42%, or around about 25,000 of the 60,000, are allogenic stem cell transplants. Allogenic stem cell transplants are the ones that we're monitoring with chimerism-based analysis. Now, in addition to new transplants, we are seeing an increase in the number of survivors due to advances in our techniques, advances including safer transplantation, availability of alternate sources of hematopoietic stem cells, expanding indications for hematopoietic stem cell transplantation, and an increase in the overall number of patients with blood-based cancers because of our increasing, or our ageing population. These advances mean that not only more transplants are being performed, but we have more survivors that need to be analysed for chimerism-based analysis. And in a recent study by MassARRAY et al., you can see on the left-hand side the increase in overall survivors. And the outcome of this study, that the number of hematopoietic stem cell transplant survivors is expected to increase 2.5 times in the next decade and 5 times by 2030. On the right-hand side, you can see that this applies to all different types of transplants, and the bottom two lines being our allogenic stem cell transplants, both related and unrelated donors. And you can see current day, we're looking at just under 100,000 survivors presently, but increasing by 2030 to 250,000. So the point here is that if you're struggling with the demand for your chimerism-based testing on STR technologies at the moment, you've got more pain to come. This is only going to increase. So let's talk about STR-based analysis. So with STR-based analysis, you take both a pre-transplant recipient and donor and analyse these for your STR markers, identifying informative loci. And then running that post-transplant sample, you're looking at those informative loci, whether it be, for example, I don't have a pointer. So whether it be, for example, it's actually a summative informative loci, which is a peak intensity that is going up, or a unique informative allele, such as you can see on the right-hand side. This is generally manually analysed, looking at those peak heights, calculating the percentage contribution for each loci, and then calculating an overall percentage contribution. Now, if you've been doing this testing, it's certainly not as clean and simple as that cartoon. There are additional challenges that we observe in STR-based electropherograms, including stutters, addition of a non-templated A. We are also, as I mentioned before, needing to analyse multiple different samples per patient, whether it be blood, bone marrow, or sorted cells, which means that you really have a challenge in throughput happening in these particular types of analyses. Some other challenges for STR-based analysis is that the window of DNA input is quite narrow. If you read the manuals for the recommended amount of input, it has a very narrow range of the amount of DNA to go into the reaction, meaning that all of these samples need to be quanted and diluted to that input range, which is quite a labour-intensive step and also prone to error. Some labs that don't dilute their DNA perform multiple injections of the sample with varying different injection times to try and factor for the amount of the fluorescently labelled products going into the capillary. And a lot of labs actually have to do both, meaning that you're increasing your cost by doing multiple replicates per sample, but you're also reducing your throughput, particularly if you're on, for example, a 16 capillary instrument. When you're looking at multiple sample types, multiple different injections per sample, means that your throughput is incredibly low. You also need to do that manual analysis to determine the informative loci, the manual calculations to determine the percentage contribution, and you're doing this on an instrument that has reducing numbers of applications. Many laboratories are phasing out capillary electrophoresis in preference for more advanced technologies for other applications. So today I'm introducing to you a more efficient panel. These issues that I mentioned before were the inspiration for us to generate a new way of doing chimerism testing for bone marrow engraftment monitoring studies. We wanted to build a panel that saves labs time and money, but also achieves the accuracy and sensitivity that you need to do this sort of analysis. So our Chimeric ID panel has an automation-friendly workflow. Standard PCR, unlabeled PCR, which can be performed on standard laboratory liquid handling robotics. We have accurate chimerism determination, determining the accurate percentage of the recipient DNA in that sample, generated by completely automated reporting. Single-click analysis that performs in seconds and generates highly user-friendly results. Taking away that manual interpretation saves you significant amounts of time and money. Now, this panel runs on the MassARRAY system. For those of you that don't know the MassARRAY system, the core workhorse of the MassARRAY system is a MALDI-TOF mass spectrometer. We use mass spectrometry to weigh nucleic acids. So that is, we're not using fluorescence. Mass spectrometry actually analyzes the nucleic acid itself and determines its mass in Daltons to determine which bases are present, and specifically, in this instance, which base has been incorporated in our single base extension. It's a robust and proven technology. It's been adopted by hundreds of clinical laboratories worldwide for a variety of different applications. It's a flexible platform, looking at SNPs, various different other types of biomarkers for pharmacogenomics, somatic mutations, and of course today what we're talking about is chimerism testing. Random access formats that you can run all of these different panels on the same instrument, in fact on the same run, on the same chip. It's a mid to high multiplex platform, allowing you to do tens to hundreds of markers on any individual sample. For liquid biopsy-based techniques, our sensitivity drives down as low as 0.1% minor allele detection. Scalable throughput from just tens to hundreds of samples per day with low-cost, simple PCR reagents, PCR followed by single base extension with no fluorescence required. So the process itself is that standard biochemistry, standard PCR followed by shrimp alkaline phosphatase, dephosphorylation of the dNTPs remaining after the PCR, and then single base extension in a multiplex format. Once you've finished your PCR thermocycling, the process is completely automated. You place that microtiter plate onto our chip prep module, which is in the center there we have an image of the system. The chip prep module is the right-hand side of the instrument. That chip prep module performs the desalting steps and the transfer onto our spectra chip. Of course, with mass spectrometry, inside that flight tube is a complete vacuum, so we transfer our nucleic acids onto a solid surface, crystallizing those with a proton donation matrix. And it transfers that chip into the mass spectrometer, does the analysis, and kicks the data through to our reporting software. All up, that time is 60 minutes from the PCR plate or the post-PCR cycling through to generating of your results, and the entire process from sample to data is performed within a single day. I mentioned we have a variety of other applications on this instrument, including liquid biopsy analysis, solid tumour profiling, pharmacogenomics, hereditary genetic testing, things like cystic fibrosis and other inherited diseases, sample identity and integrity, and today's major topic, chimerism. Now, of interest, it was in 1968 that the first non-cancer bone marrow transplant was performed. This transplant was performed in an 8-year-old boy who had SCID, severe combined immunodeficiency. SCID's not the focus of today, but it is an area of great interest to Agena. We have recently just finished development of a combined spinal muscular atrophy and SCID panel. And we're looking for early access sites to partner with us to evaluate the performance of that panel. So if you're also doing SMA and SCID testing, I would love to have a discussion with you over the course of this meeting. But let's continue with chimerism testing. And let me tell you a little bit more about the panel itself. This is a SNP-based genotyping panel. There is 92 SNPs in the panel. High minor allele frequency across all populations, so minor allele frequency approaching 50%, across all major different ethnicities and populations. Now, with a 50% minor allele frequency and 92 markers, on average we have 57 informative markers between the two individuals, the recipient and the donor, with a 99% confidence interval that we will have anywhere from 32 to 83 markers in a 99.9% confidence interval between two unique individuals. And what we're doing in this analysis is we're looking at the skew of the post-transplant sample from the normal distribution of pure samples. On the right-hand side, the left-hand side, 57 is our theoretical calculation of informative markers. On the right-hand side is our actual experimental calculation of informative markers in real samples, and you can see that that peak is clustering around the 57 marker point. So let's talk about informative markers with SNP-based analysis. So with SNPs, we are only looking at 2 alleles. All right, these alleles in mass spectrometry have unique positions in the spectra, so that is they have a unique mass. So what I'm showing here, there are 3 peaks annotated. The peak on the left-hand side is the primer if it has not been extended. Then the peak in the middle is the primer plus for example, a C base at that SNP, and on the right-hand side, a primer plus a T base for that SNP. So in what we call an informative type 1, donor and recipient are homozygote for alternate alleles. All right? And this happens 25% of the time, such that you have 1 allele from the donor and 1 allele from the recipient in your analysis. We also have an informative type 2. Informative type 2 is where we're looking at where the donor is actually homozygote, and if the recipient is present, we're seeing the emergence of the alternate allele where the recipient was heterozygote. So that's what we call an informative type 2. And an informative type 3 is the opposite. The donor is heterozygote and the recipient is homozygote, so we're looking at a summative allele, meaning that we have contribution from we have the 50-50 allele from the donor and an increased contribution of one of those alleles from the recipient. Of course, the last, approximately 25% of the time, are markers that are uninformative, where both donor and recipient are homozygote for the same allele. Now again, that is a simple cartoon. Let's have a look at see what it looks like in the mass spectrum. So here what I have is, on the left-hand side, individual A and individual B. Individual A with that blue annotated peak is homozygote, for example, for the C allele. Individual B, homozygote, for example, for the T allele. You will see a grayed out allele in the middle. That's from an alternative assay in the multiplex. And on the right-hand side, you can see the varying different contributions when we mix these samples together. At the top, a 10% of sample number B, 5% through to 2% down the bottom there. And what we're doing is we have a normal distribution of wild-type samples that's built into the software. So we know what the allelic frequency is for homozygote, homozygate, and heterozygote in a pure sample. And in our post-transplant sample, we're looking at a skew of those alleles. So the normal distribution allows us to calculate a Z-score for each assay. And then we look at the informative assays and determine whether there's been a statistically significant Z-score event in each individual assay. Now, I understand that sounds quite complex and probably just as complex as STR-based analysis, except that you don't do any of this. The software does this for you. These are digital signals that have a very clear baseline. You're not looking at any of this, you're not determining informative loci, you're not doing any calculations. You are clicking a button and the software does all of this for you. So let me walk you through the user experience, because this is the detail under the hood, because I know you're scientists and you all want detail. But let's go into more of the user experience. So the first step, of course, is logging in your samples. So we have a very simple user interface to log in your samples, giving them a sample ID, identifying whether they're a pure recipient, a pure donor, or a mixed, a non-template control, or some other form of control. And we also identify we assign a sample group, such that the software knows that this donor and this recipient is associated with this post-transplant sample and multiple post-transplant samples. So that donor group is then used to do the analysis further down, identifying those informative markers and doing the calculation. And what we've also built into this system, because of course this does happen on a frequent basis, it has the capacity to do multi-donor analysis, where you may have had an initial donation that has failed and then gone through a secondary donation, and the software can have multi-donor profiles in there and calculate the percentage contribution from each potential donor. So once you've identified those samples and assigned them to the group, the raw data is passed from the mass spectrometer into our software algorithm. It references those pre-transplant profiles to identify the informative loci, and it also verifies the various different QC criteria to ensure that you've had a highly informative and accurate result generated. Ultimately, it then outputs a report with the percentage recipient and the percentage donor contribution, or the sample composition. So let's have a look at what those reports look like. On the left-hand side is a very simple single sample report. So what you have here is at the top there you have your patient group identifier, your sample ID. On the right-hand side, whether it's been called as chimeric or not chimeric, the various different Z-scores and statistics underneath that, and then you can see the percentage donor contribution at 83.6% and the percentage recipient contribution at 16.5%. Now that's just for an individual post-transplant sample. But usually what you're doing is you're tracking post-transplant samples over time. So on the right-hand side is a historical analysis. So that is, in that patient group ID, you may have multiple post-transplant samples, and it's reporting what the percentage chimerism was for each of those different time points. Time point number 1, time point number 2, time point number 3, time point number 4. So you can see that if there has been a change over time in that particular individual. So two very simple and user-friendly reports One which is looking at that particular sample that you've run today versus an alternative one where it's giving you a historical analysis. Now, of course, you don't run the pure donor and recipient at the same time as every transplant sample. That's run prior and it's stored in the database, right? So it's not something that's an on-plate analysis. So when we looked to demonstrate the performance of our new panel, there were a variety of different characteristics that we wanted to demonstrate. And in the following slides, which will summarise a series of experiments, I'm going to show you our accuracy, our reproducibility, our DNA input tolerance, our limit of detection, and our comparison to STR-based methodologies. Now, before we do any of these studies, we ran into a challenge. And the challenge was determining a reliable source of truth. Now, we could take clinical samples that had previously been run by a single STR-based analysis, but how sure were we of that truth? And in fact, the confidence interval around that truth was larger than the level of quantitation that we were trying to demonstrate. So that became a significant challenge. So we needed to generate a model system. And we did that by taking male and female samples, such that when we mixed them, we could use deep digital PCR of the X and Y chromosomes to determine what the truth composition of that sample was. So for all of the studies that I'm going to show you, that's how we made these contrived samples. And in later studies, I'm going to show you real clinical samples as well. But to prove our accuracy and reproducibility and so on and so forth, we use this model system. All right, so the first study that I'm showing you is in the area of accuracy. This is a single-site accuracy study. We made 2 independent dilution series for 2 different sets of individuals. We performed 8 different replicates per dilution point. Of course, we use that digital PCR on the XY analysis to determine the truth composition. And our dilution points range from a composition of 2.5% through to 45% chimerism. And what you can see in the graph on the right-hand side, we've got excellent linearity with a slope of very, very close to 1, an R-squared of 0.99, and an average variance from the digital PCR of 0.8%. Our median variance from the digital PCR results was 0.48%. And therefore our coefficient of variance is 1.83%. And that is a true percent. So where digital PCR might have said, okay, the percentage contribution is 50%, then the variance that I'm stating here as 0.88% is from 49.2% to 50.8%. Okay, just to make sure we're clear on that. So excellent accuracy in a single site, but the truth of the test of a technology is not whether the development lab can get it right, it's whether outside laboratories can get it right. So here I'm showing you a multi-site, multi-user accuracy study. So that is, we selected 3 locations, 3 different instruments, and 3 different operators. So, and this wasn't just a laboratory next door. We went global. You've picked up my accent by now, I'm sure, right? We sent a sample to Australia, right? We sent a sample to San Diego, we sent a sample to Europe, right? 3 globally diverse sites. 3 different instruments, 3 different operators. We sent 3 different samples, a 3%, a 5%, and a 20% chimerism sample, and there were 13 replicates performed per dilution site, and of course we have that XY ID analysis. And what you see on the right-hand side is that accuracy is reproduced in our multi-site study, an average variance from the digital PCR result of only 1.3%. So highly reproducible, even when we send this all around the globe. All right, to further look at reproducibility, we've done a further study, 2 independent dilution series, 8 replicates per dilution point. This is similar to the study that I showed you before, but instead of showing you a scatter plot with a broad dynamic range, I'm now zooming in on the reproducibility. So let me explain how we've done this. So we have samples going from 2.5% or 3% through to 50%. But what I've done is I've zeroed out the truth value. So that is because if I show it to you on the scatter plot, you can't see the real variation. right? So where the digital PCR said it was 3%, I've set that as 0. Where the digital PCR said it was 50%, I've again set that as 0, and we're looking at the variance around that 0. So you can see it on a finer scale. So what you can see here is regardless whether it's a 3% chimerism or a 50% chimerism, the variance around the truth is similar. So we're seeing a standard deviation falling within plus or minus 1%, rather than whether it's 3% chimerism or 50% chimerism, with the largest variation being plus or minus 1.8%. So highly reproducible results regardless of the percentage contribution of chimerism. Now, with STRs, DNA input is the significant challenge, as I mentioned before. Too much or too little DNA causes a variety of different challenges. The quantification can be off-scale and the calculation not accurate. You see an increase or a pull-up of stutter peaks, poor spectral separation can be observed, incomplete anucleotide addition, and allelic dropout on the other end when you have too little DNA added in. So laboratories do one of two things, and often both. They either dilute the DNA before PCR, normalizing every single sample into the correct input, or alternatively, multiple CE injections with multiple different injection times. And often, both of these are necessary. So we wanted to show whether our panel was accurate regardless of the DNA input, and that's what I'm showing you on the right-hand side. On the right-hand side, same samples at 2%, 6%, and 12% were inputted into the reaction at 2 nanograms, 10 nanograms, 100 nanograms, and 200 nanograms. And regardless of the input amount, there is no statistically significant trend here. The results are accurate regardless of your DNA input, meaning that you don't have to dilute your samples anymore. You don't have to do multiple injections. We don't have injections in mass spectrometry. So you can just take your extracted sample and immediately put it into the PCR reaction without quanting or diluting. Now, let's look at the limit of detection. So in this study, we have focused in on the range of low-level chimerism. Samples at 1%, 2%, 3%, and 5%, and of course pure being 0%. Now, our standard default settings that are built into the software is we only report chimerism if it's detected above 1%, a 1% frequency, and we only report it if it has a Z-score of 2 or more. These settings are user adjustable. We have focused these settings on accuracy, such that these settings, as you can see in the pure, of the 48 pure samples that were run in this study, none were reported as chimeric. So we have focused our settings on specificity and not the trade-off of specificity, which is sensitivity. But still, we are achieving 100% detection of our 3 and 5% chimerisms, 83.3% detection at our 2% chimerism, and 16% at 1%. Sorry, bear with me for a moment. This is a tough voice to keep going. All right, so now we wanted to do a comparison with STR-based analysis, because everything I've shown you so far has been using our model system with digital PCR XY analysis to determine truth. So with the Brigham and Women's and Children's Hospital in the United Kingdom, not the one here in this country, we analysed some quality assurance program samples. This is a UK-based quality assurance program where they have more than 100 participants. The way the program works is that participants receive 2 samples per trial with 5 trials per annum. And out of those 100 participants, what is calculated by the trial organisers is a robust mean, so that is the mean chimerism result generated by all participants, and also a robust standard deviation. So, of course, you know what a standard deviation is. What they call a robust standard deviation is a pass if you're within a plus or minus 2.5 standard deviation from the mean. So this is the way the quality assurance program is run. So the way I'm showing these results, again, I'm zeroing out I'm zeroing out the robust mean. All right, so these, you can see what the actual percentages are up the top, first one being 8.5% on the left, the second one 11.5%, 79%, so on and so forth. I'm zeroing out that expected robust mean so that you can see the true variance around the robust mean. This is the only way I can think of to show you data that is actually meaningful. Right, so the 2.5 standard deviation around that robust mean is the thin black line. So basically, as part of this quality assurance program, if laboratories report results that are within the range of that thin black line, then that is considered a pass. And then I'm showing you the Birmingham result on their STR analysis and the Birmingham result on the Chimeric ID analysis. So the STR being in red and the Chimeric ID being in white. So for that first one on the left-hand side, it looks like the STR was around about 0.8% lower than the robust mean, and the MassARRAY, I think maybe 0.9% lower than the robust mean. Highly concordant there when STR and Chimeric ID are run on the same sample at the same site, so the same tube aliquot. So every sample here is within the robust mean, so this is a pass. But you can see that they are highly clustered around the 0 point, which is our expected, which is the every sample is within the robust standard deviation. Let me start that again. Every sample is within the robust standard deviation, but you can see that they're very close to 0. And when you look at the comparison with STR done at the same site, there's high levels of concordance. So what does this mean for you? It means that the Chimeric ID panel is highly concordant with STR-based analysis. So patients that you are currently monitoring with STRs can be ported over to this new panel without you having to go back and reanalyze anything except their pure and recipient, their pure donor and pure recipient result to put that into the database to determine new informative loci in the Chimeric ID panel. All right, so that's an external quality assurance program, but in addition to that, Agena commissioned our own internal quality assurance program. So what we did here is we generated a variety of different samples at 5, 10, 15, 20, and 25% chimerism confirmed with our digital PCR technology. We sent those samples to 3 different STR sites, again global, all right, again all the way around the globe. So we sent them to those STR sites and they ran them in duplicate. So we've got 6 different STR values which are shown to you as the blue dots. And then we have the Chimeric ID result which is shown to you in the red dot, as the red dot. So you can see that we're highly concordant with these various different STR-based different analyses, except for that one funky blue dot up the top there. I do not know what happened there. Maybe just ignore that one for now. Obviously some aberrant STR result happened there. But you can see that the Chimeric ID is highly concordant on these samples with multiple different STR technologies at multiple different sites. So again, that's it's an internally or Agena commissioned quality assurance study, QAP study or proficiency study. So what I've shown you today is that the Chimeric ID panel shows excellent accuracy and reproducibility when we compare it to our method of determining truth, which was our XY digital PCR analysis. Dilution of your DNA is not required, as performance of the panel is consistent across a very wide range of DNA input. The panel shows high levels of concordance with STR-based methods, so that means that you can convert technologies without affecting your existing patients that are being tracked via chimerism, and a completely automated data analysis, meaning that you can save significant amounts of time, and we all know that in this business, time equals money. So thank you for your attention today. I'd be pleased to take any questions. Okay, so the question is, can I comment about the methodology of MassARRAY technology versus capillary electrophoresis? So with the MassARRAY, what we're doing, we're doing multiplex PCR amplification of those 92 different regions. Right? So we're amplifying the region of interest, and then we're doing single base extension. So we're annealing a primer adjacent to the single nucleotide polymorphism, and we're extending a single base. All right, for each of these. They're all multiplexed in the reactions, but that's what's happening in at each individual loci or SNP. So what's happening there is that we have a sequence for that primer. Right? We know its mass, it might be 5,000 Daltons, and it's extending onto a C, which will be 5,289 Daltons. Or it's extending onto a T, which will be 5,369 Daltons. It's taken me 10 years to remember these numbers. Right, so we've then got those two possible alleles. And then what happens is that in the mass spectrometer, the laser itself is ablating the so the biochemistry is generating the primer plus a single base. We know the mass of those potential alleles. We're spotting that onto a spectra chip and introducing it to the mass spectrometer. The mass spectrometer via a laser is pulsing, ionizing, flying those molecules and detecting their molecular mass. I actually think of it as capillary electrophoresis, but in the gaseous phase, right? Because what happens is the smaller molecules fly faster than the larger molecules, but not based on the number of nucleotides in them, but based on the molecular mass. So we're not pulsing that laser one time, because one analysis takes 3 nanoseconds. From pulse, ionization, time of flight to detection, 3 nanoseconds. Right, done. So what we actually do is we pulse that laser hundreds of times, right? And if you do an experimental measurement hundreds of times, what does it mean? You become highly sensitive and highly quantitative. So that's what we're doing with the mass spectrometer, is we're actually taking hundreds of analyses in order to get the frequency of the alleles at that particular position. So a little bit like capillary electrophoresis, in that the small molecules are moving faster than the large molecules, except it's only taking 3 nanoseconds to do it, so we do it hundreds of times, because we're now in a gaseous phase. Hopefully that's mass spectrometry in a nutshell. Well, yeah, so we've shown that we have similar accuracy to the various different STR-based analyses, which I showed in several of those slides, and we have a similar limit of detection as STR-based technologies as well. That was that was table stakes for us to develop a chimeric ID panel. We just had to have accuracy and sensitivity. The value add is the completely automated data analysis and no dilution and other things.
Are you doing multiplex PCR to amplify all the SNPs in one reaction?
It's actually in 8 reactions. So it is spread across 8 reactions. The panel is 92 SNPs, but spread across 8 reactions. But generally, you've got more than enough DNA. Unlike oncology, which we talked about 2 hours ago, where you don't ever have enough DNA, in these sorts of analyses, you have more than enough DNA. Where's Keith? So the instrument itself is around about the sort of 150,000. We have multiple different configurations, so it depends on your throughput. We have a 96 configuration, a 384 configuration, but generally it's around about the $150,000 mark for the instrument that can run all of these various different applications. So if you come via our booth, in our booth we have a do the math calculator, which allows you to put in all of your details, the number of samples you're running, your existing methodology costs, and you can calculate the savings for your laboratory by this instrument, even including the purchase of the instrument.
So I guess I missed the beginning part. What is the maximum link that you can sequence accurately with those Daltonic weight differences?
The maximum length?
Length of the sequence.
Okay.
So, because the difference is going to be really minimal when you add to the length of the sequence for the time of flight.
Yeah, well, we're really only sequencing that one base, right? So we have a primer that anneals adjacent to the single nucleotide polymorphism, and it extends onto one, the other, or both allele that can be present there. So we're, unlike STRs where you have varying different sizes and everything else, what we have is we're just sequencing a single base, but we are multiplexing it because each primer has its own sequence, so it has its own mass in the multiplex, and each primer plus each of the potential alleles that can be present has its own unique mass. See, we're measuring down to 1 Dalton. A hydrogen atom is 1 Dalton. 1 proton or 1 neutron is 1 Dalton. So yeah, our spectral separation is a lot finer than you would see with STRs.
Okay, thank you.
We use, so this panel is in 8 wells, so eight reactions, but it's still cheaper even with 8 reactions. These are unlabeled unlabeled primers. You know, our polymer is actually a vacuum tube. It's pretty cheap to run a vacuum, and so it's still cheaper for those 8 reactions. The benefit is, if you were to have a failed reaction, it doesn't mean that you actually are lacking a result, because the markers are spread across those 8 reactions. Now, we don't want to have failed reactions, of course, but if you were by chance, and the low probability that that happens, it won't mean you get a null result. You still have enough informative markers across the all of the other wells, so you don't have to repeat. Sorry, I missed that question. Yes, so if you come down, I can assure you we will show you a lower cost solution with our Chimeric ID panel. If you come down to the booth, we can show you the full calculation with our do the math calculators. All right, well, I don't know about you, but I've had a solid day. So I think this is the last workshop of the day, so I hope you enjoy your evening. Please do, there are forms in front of you. If you fill out those forms, we can send you more information. We do have a white paper which describes everything that I've talked about today in our various different verification, validation, comparison studies that we would be happy to email you. If you fill out the form, drop by our reception desk at the front or come to our booth during the meeting. Yeah, we can send you more information.